Thank you. It's now my pleasure to introduce Dr. Laura Evgin, who was selected as a 2024 Canada Gairdner Early Career Investigator by Dr. Sadelain. Dr. Laura Evgin is a scientist at the Genome Sciences Department at the BC Cancer Research Institute and an assistant professor in the Department of Medical Genetics at the University of British Columbia in Vancouver. She earned her PhD in biochemistry from the University of Ottawa under the guidance of Dr. John Bell, followed by a postdoctoral fellowship at the Mayo Clinic in Rochester, where she worked with Dr. Richard Weil. In 2020, Dr. Evgin established her lab at the BCCRI, focusing on enhancing the efficacy and accessibility of T-cell therapies for cancer. Her lab leverages diverse tools and model systems to design innovative CAR constructs, develop CAR T-cell engineering strategies, and evaluate CAR T-cell function in immune-competent models of cancer and autoimmune diseases. Dr. Evgin will be presenting Engineering Effective and Accessible CAR T-Cells. Please join me in welcoming her to the stage.
Last speaker was a little bit taller. Okay, so good morning. Thank you very much. This is a real honor to be here today. I'd just like to start by congratulating Dr. Sadelain and Eshar, as well as all of the other laureates on their award, and thanking them for their really important contributions to science. Okay, so T cells patrol the body searching for signs of infection or cancer. They do so using their T-cell receptor that interacts with the MHC molecule that is expressed on virtually every cell in the body and express small bits of peptide of the proteome. When a T-cell encounters its cognate ligand, it produces cytokines and other cytotoxic molecules that enable cell lysis. And together, it's really this specificity, functionality, and memory properties of T-cells that allow them, or that endow them with very strong agency to recognize and eliminate cancers over time. Importantly, we can also help T-cells to focus their attention on cancer cells by engineering them with this synthetic receptor called a chimeric antigen receptor, or a CAR. So as Dr. Sadelain just outlined, the CAR brings together the recognition capabilities of an antibody with the signaling properties of a T-cell receptor. The first iterations were developed by Dr. Zelig Eshar and fused the variable heavy and light chains of an antibody with the constant TCR alpha and beta chains of the T-cell receptor. This was then simplified to directly fuse the scFv to CD3 zeta. And in both these configurations, a T-cell expressing these new receptors was conferred a new specificity for surface-expressed antigens in an MHC-independent manner. However, because effective T-cell responses require not only signal 1 in the form of TCR stimulation, but also signal 2 in the form of co-stimulation, it was really the addition of this co-stimulatory domain by Dr. Sadelain and his team that overcame the apoptosis and dysfunction that was associated in primary T cells, and really turned this into a therapeutically useful molecule. They also selected CD19 as a target antigen, and this was important because it enabled the clinical translation in the absence or with a manageable toxicity, on-target, off-tumor toxicity profile. So together, the development of the CAR and was really opened up this field of synthetic biology and synthetic immune receptors, and provided the motivation to develop manufacturing and regulatory frameworks for engineered cell therapies. So CAR T cells have been enormously successful for hematologic malignancies. There are now 6 FDA-approved products targeting CD19 or BCMA for the treatment of acute lymphoblastic leukemia, large B-cell lymphoma, and multiple myeloma. CARs targeting CD19 are now an established therapeutic modality. They can induce prolonged remissions in patients with B-cell malignancies with minimal long-term toxicities. Although they were initially approved in the third-line setting, it's now been shown that they can be an effective alternative to autologous stem cell transplant in the second-line setting for patients with early relapsed or refractory large B-cell lymphoma. There's also now more than a decade of follow-up that has elapsed in some of the early patients that have been treated, and it's really remarkable to see that some of the early patients who were treated still have circulating adoptively transferred cells in their body that continue to protect against relapse. So the overarching goal of my lab is to increase the accessibility and effectiveness of CAR T-cell therapy for both oncology and autoimmunity. There are many levels of scale over which to design and optimize these synthetic receptors that are engineered into T cells and which need to operate in both the micro and macro environment of an individual with cancer or autoimmune disease. And to that end, we've invested in the development of immune-competent mouse models in order to reverse translate some of the clinical observations, as well as assess complex cellular dynamics in the setting of combination therapies and evaluate novel ways to engineer these CAR T cells directly in the body. So there have been many clinical correlates of response in the setting of large B-cell lymphoma, and I'd just like to highlight one, and that is the presence of extranodal disease. So as you can imagine, lymphoma primarily exists in lymph nodes, but in about 1/3 of patients, the disease occurs elsewhere in the body. This can either be as a primary event or as a manifestation of advanced disease. And so there's now several studies that have retrospective studies that have documented that patients treated with axi-cell or teasa-cell and who have substantial extranodal disease burden fare worse than those who have fewer or no sites of extranodal disease. And we're looking to recapitulate this in our mouse models of lymphoma. So when we treat our mice that have lymphoma with either 4-1BB or CD28 co-stimulated CD19 CAR T cells, we can dramatically reduce the levels of malignant B cells in these mice. But at this dose, we see that the mice ultimately succumb to disease. At necropsy, we see that the disease is relatively well controlled in the lymph nodes and the spleen, but we see substantial disease burden in the liver and in the ascites. So over time, the efficacy of B-cell clearance varies across anatomic site, and this is shown in the CAR T-cell to B-cell ratio. Particularly, there's this difference between lymphoid and non-lymphoid tissues. And this has led us to hypothesize that there are different barriers that exist within these anatomic sites which underlie differences in functionality of the CAR T-cells. And so to explore this, we've taken on two different approaches. The first is to design several spectral flow cytometry panels to characterize the tumor microenvironment as well as phenotype the CAR T cells over time and across anatomic sites. And we've seen this very rapid tissue imprinting on T cells. We hope to use this information in order to better arm CAR T cells to perform better at these sites of refractory disease. We've also taken on an unbiased approach where we conducted an in vivo CRISPR knockout screen using a small panel of 120 transcription factor guides to identify genes to either knock out or overexpress in CAR T cells to enhance their function. We've identified a number of candidates that we think would globally ameliorate CAR T cell function and some other ones that we think may be more site-specific, and we're in the process of validating these different candidates. Stemming from my work as a postdoc, we're also very interested in how we can leverage the native T cell receptor in CAR-modified T cells in order to enhance the function, engraftment, and persistence of these cells, particularly in solid tumor models, now using an mRNA vaccine. And so in our system, we have OT1 transgenic T cells spiked into a wild-type pool, transduced with an EGFR-v3 CAR, and these cells are then boosted in vivo with an OVA encoding mRNA vaccine. And I'll just point out that our tumor model does not have OVA in this, so this is an irrelevant antigen. Provision of the vaccine is able to substantially increase the global level of circulating CAR T cells in the blood as well as in the tumor, and these OT1 T cells really overtake the population. We also elicit a shift in the functional profile, specifically in the OT1 CAR T cells. There we go. I just wanted to point out that although the CAR was designed in order to mimic the TCR, we think that this is a really interesting way to engage alternative signaling, alternative differentiation programs and signaling in order to maximize their effector function, particularly in the solid tumor setting. So CAR T-cell therapy has really shifted how we think about treating CAR T-cells at the global level, how we think about treating disease at the, with engineered cells globally. There are more than 1,600 trials listed on clinicaltrials.gov. The vast majority of these are for cancer indications. However, in recent years, there's been intense interest in using these cells for autoimmune diseases, where CD19 and BCMA targeted CAR T cells have been repurposed to treat diseases where B cells and plasma cells elicit autoimmune pathologies. And so we're also very excited about this, and we're collaborating with a number of different groups at UBC in order to apply our CAR T cell expertise to mouse models of systemic lupus erythematosus, MS, and type 1 diabetes. So all of the commercially approved CAR T cell products and a majority of cell therapies that are undergoing clinical testing are made from a patient's own T cells ex vivo. Very, very briefly, the T cells are collected via leukapheresis. They're stimulated in vitro, transduced with a viral vector encoding the CAR, and then expanded to generate a engineered cell product comprising hundreds of millions of modified T cells. As you can imagine, this is a complex process, and the list price for the commercially approved CAR T cell products surpasses $500,000 US. This creates an equity and accessibility barrier and has significantly slowed implementation, particularly in the Canadian healthcare system. So this has provided the motivation to develop strategies to directly generate these cells in the body by direct infusion of either lipid nanoparticle encoding lipid nanoparticle formulated nucleic acids or targeted viral vectors. And we have been collaborating with a number of groups at UBC in order to optimize the design elements for an LNP RNA delivery system for this CAR instruction. At the foundation of this, we think that we need a delivery system to efficiently transfect T cells or other effector cells. And to this end, we've been collaborating with Dr. Peter Cullis and Nanovation to use their long circulating LNP formulations, which have enhanced transfection of immune organs and reduced reduced hepatic tropism. We're also interested in how we can optimize the duration and intensity of expression through mRNA cassette design optimization and choosing ideal RNA species. Finally, at the top of this mountain summit, we're also interested in how we can optimize the payload through CAR design in order to choose designs that will maximize CAR T cell functionality over this very transient expression period. So to conclude, I'd just like to thank the members of my team at BC Cancer and UBC, our many collaborators, because this really is a team effort, and as well my past and current mentors. So thank you very much.